Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest who has been involved and continues to create a better tomorrow for so many people out there. Today, we have the honor of being joined by Dr. Carl Dieffenbach, who is the director of the Division of AIDS at the National Institute of Allergy and Infectious Diseases, part of our National Institutes of Health here in the United States. Dr. Dieffenbach has oversight of the global HIV/AIDS research portfolio, over $1 billion a year, and a staff of over 150 federal employees, and is responsible for the planning, the implementation, managing, and ultimately evaluating programs in fundamental basic laboratory research, the discovery and development of therapies and various treatment strategies for HIV infection and its co-infections, and its complications through basic research as well as clinical trials, as well as the discovery and the development of vaccines, topical microbicides, and various other prevention strategies through their research and clinical trial programs. Over the years, Dr. Dieffenbach has played an extremely instrumental role in restructuring the Department of AIDS-supported clinical trials research networks, has actively fostered collaborations, partnerships with various other federal agencies, international research groups, professional societies and foundations, as well as industry and community advocacy groups. Dr. Dieffenbach received his bachelor's degree in biochemistry from University of Maryland, did his PhD in biophysics at Johns Hopkins, and completed his postdoc work at the Uniformed Services University of Health Sciences and the Department of Pathology. A lot of really interesting themes to get into today on an extremely timely topic. But Dr. Carl Dieffenbach, thank you so much for taking the time to come on the show this morning.
It's great to be here with you, Ira.
Great having you, Carl. I would love to start off. I know we're going to be talking a lot about HIV today, but I would really like to start off sort of a few years before you got involved in this space, because I was taking a look back at some of your doctoral work and the postdoc period where you're working on the regulation of interferon synthesis, induction action in the early 80s, which proved very important, not just in infectious diseases, but oncology and in other areas. Take us back to the early days, sort of pre-HIV, if you would, and a little bit of what got you interested in this whole area of biology, because it's extremely fascinating how it leads into what you did next.
So just a very short story. Something that got me really hooked on science was a lecture by my freshman chemistry professor where he showed a Flash Gordon cartoon where Flash was traveling through the universe and got this horrid infection. And the doctor on the flight purified this magic compound called interferon and saved Flash's life. And Dick Goldsby, who was the lecturer, said, interferon is real. And I thought, well, that's one of the coolest things I've ever heard. I want to study that. So I followed him back to his office and didn't leave him alone until he let me work in his lab. I've been hooked on not viruses per se, but how the human body fights viruses it's pretty much my entire career in a nutshell.
No, that's awesome. And it's I think it's in the late 80s, maybe 1990, where we see that theme of interferon and HIV sort of come together in right. A paper that you write. I think you're at the Henry Jackson Foundation at the time. And these are the very early days of the HIV epidemic. And needless to say, you've been there since then. And you've been on this journey that has seen a death sentence become an extremely livable condition, and what we'll be getting into, potentially something we're going to completely eradicate. Talk a little bit about that time and a little bit about this transition that you have seen since the late 80s to 2023, and this amazing sort of unmet medical need that we have, built such an amazing therapeutic armamentarium around.
So the paper you're referring to was done with a guy now who's at the University of Nebraska named Howard Gendelman, where we looked really carefully at macrophage as they become infected with HIV in cell culture, and realized that they were the infection actually caused a defect in interferon production. So that paper was published in either 89 or 90. That effect has been rediscovered several times since, because nobody ever reads the old literature. But when I had formed a very close personal mentoring relationship with a faculty member at Uniformed Services named Peggy Johnston. And Peggy took an interest in me as a graduate student and then as a postdoc, and said, we could really use you in HIV research. She had gone to the Division of AIDS. So I said, let me come out and talk to you about where I am in my career. This was early 1992. And she said, I thought you would be a great program person, but you have grown so much. Will you consider joining as a branch chief? And so I applied and got the job. And Peggy was my mentor all those years. And immediately I got heavily involved in making new antivirals, because that was what we were all about at that point in time, is we had a disease that we could treat the consequences of frank AIDS. We had antibiotics to treat Mycobacterium avium, to treat TB, to treat crypto, but it was all really crude stuff. And in the meantime, we had AZT at very high doses, which was causing all kinds of consequences for people. And immediately, with understanding the growth of the virus, we realized that resistance was cropping up. A guy named Doug Richmond at UCSD really propagated this idea that in order to truly treat HIV, you have to stop it cold. And this was a really novel idea. At the same time, a group out at Frederick, led by Jeff Lifson, published a paper using PCR, which is a technique I had been really experimenting with, basically saying the virus doesn't go away. It replicates every day, every minute of every day, constantly in somebody who is living with HIV. And the viral load was being measured at levels of 10 to the 4th, 10 to the 3 to 10 to the 4th to 10 to the 5th copies of virus per mil. And it was like an eye opener, just how important therapy would become to stop the virus cold. So we then had, in retrospect, we had a vision of what the problem was. And David Ho said it best all these years ago: it's the virus, stupid, for which he remains famous for. But through the collaborations that the division had, and that's where this whole notion of collaboration started, is we had these programs that partnered academics with industry to explore drug discovery and development. And from those programs grew better nucleoside analogs, grew the first generation of protease inhibitors, to a point where then the first really successful one was Crixivan from Merck. We were not really involved in that, but we did ultimately help with some of the evaluation of 3 drug combinations. And then in 1995, in very quick order, I joined the division in 92. Between 92 and 95, we had the first 3 drug combinations that demonstrated efficacy and what many people called the Lazarus effect. People who had almost no CD4 cells, literally were emaciated, were literally coming back to life. Their CD4 count would jump up to 300 or 400. They would gain weight. and literally be able to rejoin society, rejoin working for a living. And this became the issue from that point on about two things: improving the safety of the drugs, reducing the side effects, at the same time access. And our first area of emphasis was access in the United States.
Mm-hmm.
But in very fast order, the industry realized they could do well by doing good, and got into drug discovery and development, and a whole next generation of different drugs came along, better nucleoside analogs. We ultimately got the nucleotide analog that became tenofovir diproxyl fumarate, TDF.
Mm-hmm.
And then ultimately there was TDF FTC that came out from Gilead. And there are all these company movements that we could spend days discussing. But ultimately the goal was a pair of nucleoside analogs with another anchor antiviral that targeted a different mechanism of action. And so it became either a non-nucleoside reverse transcriptase inhibitor or a protease inhibitor. But at the same time, viral load was coming along. And we could tell that their viral loads would be detectable. They would start these medicines. And both George Shaw and David Ho showed that these drugs were literally stopping the virus cold. So at the individual level, we had this sense that these would have a profound effect. As we got into larger and larger amounts of people on treatment, the CDC noticed a decline in deaths. Mm-hmm. And we really had then a model of where treatment could have a profound impact. And throughout the late 90s, the drugs kept getting better, and we kept trying to tinker with the regimens. But then the game changer came in the early 2000s when Dr. Fauci had the opportunity to talk to George W. Bush and say, we need to do that, because Laura had been to Africa and seen all these children dying. Yeah. And she came back and it was like, we need to do something. And questions were asked about why aren't the drugs in Africa? And there were people that didn't believe that the continent was ready. I mean, it's so colonial and so painful to talk about this time. But really, it was about the belief that, give educated people the opportunity and they will do what they can do. And Tony, working with people in the White House, working with people ultimately like Mark Dybul, who was on his staff, put together the PEPFAR program. Mm-hmm. That then ultimately got us to where we are today with 25 million people around the world on therapy. The reduction in death has continued through the delivery of antivirals. And it is probably the most important scientific breakthrough, both in terms of discovery and then global implementation of the late 20th century. Mm-hmm. And we were there every step of the way as an institute, either supporting or pushing or cajoling or working with the Congress, working with others to make these drugs available. I can't say enough about the Clinton Health Care Foundation that really helped negotiate. Ira Magaziner came in and helped negotiate reduced drug prices that got it to a point where now for about $100, $150 a year, a person in Africa can have a year's worth of the highest quality antiretroviral therapy. So we've seen a sea change. We've been part of it. Now, beyond that, that's like the top line message. Right. But in addition to that, these drugs became so good, we looked at them for the notion of treatment as prevention. Yep. So Mike Cohen and his team at UNC had this brilliant idea to say, if you had discordant couples, 0 different couples, and you put the person living with HIV on therapy, reduce their viral load to undetectable, would there be HIV transmission? And this study was called HPTN 052. And through this analysis of couples and careful monitoring of viral load, the 052 team showed that if you are virally suppressed, you can't or you don't transmit to your partner. And then there was an extension done where we maintained people on therapy, and there were still no transmissions if the virus was undetectable. And this led to, I think, one of the most important behavioral interventions we've had in HIV in a very long time is the U equals U movement,
right. Yep.
Where undetectable equals untransmittable, and essentially you are freed from your guilt. If you are living with HIV, you cannot transmit to a person that you love, and you are free to be you. You're free to love who you want, when you want, how you want, but you've made a pact that you're going to take your medicine on a daily basis. So this really becomes a life-affirming process. So we helped make the drugs, and then we also helped define how the drugs were used. And I could go on and on because there are other uses for the antivirals that I think are very important. Mm-hmm. But I think that's what you were asking for, some sort of a big picture view of where we've been and where we are today when it comes to use of antiretrovirals.
Absolutely. Yeah, and let's continue down that path, because you were just saying we're now blurring, the treatment prevention domain. You mentioned undetectable equals untransmissible. We've hosted some of the Gilead folks in recent months per the concept of pre-exposure prophylaxis using these interesting cocktails, and then within that prevention portfolio exist other things, and we'll be getting into the vaccines, of course, which are an extremely important topic now. Also, we profiled one of the City of Hope patients, patient number 4, per the immune system reconstitution per the CCR 5 delta 32 mutation. Give us before we get into vaccines, because this is going to be sort of the core where we're going with this, but give us sort of the overview of the prevention basket as it exists today within your portfolio at the Division of AIDS, and sort of what the priorities are in that basket. Put these things in context for us to get a perspective on. It is within our vision now that this thing may be eliminated completely. So, it's just, we don't hear this with dementia or cancer or other, but here is a case where you're beyond treatment now. And so I think it's extremely important just to talk about this portfolio at NIH today.
So before we go on, therapy is great, but it's still one pill once a day. Yeah. And there are people that cannot abide that. And so we are still working on improving therapy by making long-acting treatment for people, because this ultimately is about choice. And it's the choice of somebody who's living with HIV for how they want to receive their therapy. But choice also applies on the prevention side. Mm-hmm. How do you approach a young man in Uganda, a young man in the rural South, a young woman also living in sub-Saharan Africa, and give them the choices for how they wish to have HIV prevention come into their lives? HIV prevention started with pediatrics, with a trial called ACTG 076, which was the use of AZT to prevent mother-to-child transmission. And the rate at the time was about 25%, because there was no treatment whatsoever. And the use of a single, relatively weak nucleoside analog reduced that level of transmission to approximately 4 to 5%. So that was a major game changer that occurred very early in the pandemic. But then we had this idea about, one of the things we are, first and foremost, is a research organization. What does a trial result tell us about that kind of a situation? And what does 076 teach us about HIV transmission? And it is, the lower the viral load, the less chance you have, the less virus you're exposed to, the lower your risk is. There was then studies done in adults in Uganda that looked at populations of, in serodiscordant couples, in serodifferent couples, and then the higher the viral load, the more likely you were to have transmission. And the Rakai team showed that for people with very low viral loads, transmission was negligible or non-existent. So when we think about this, how can you reduce the amount of virus somebody's exposed to? We then went and did two trials to complement then a French trial on adult medical male circumcision. And this has been one of the major advances early on in prevention, that for heterosexual transmission, at the time, there was an almost identical rate between male to female and female to male in a non-circumcised population. Okay. Once the men are circumcised, their rate of acquisition drops precipitously. So this sets off all kinds of political debates, as you can well imagine. Sure. But again, PEPFAR and others have gone and taken those findings from the early 2000s and implemented adult medical male circumcision, voluntary adult medical male circumcision throughout sub-Saharan Africa. And that's been a major tool that has been used. So again, choice. So that's one choice. We, fast forwarding to the use of pills, again, we knew that AZT worked for the purpose of mother-to-child transmission. We tried to do better in the mother-to-child transmission space by adding other drugs. And slowly but surely, we've pushed those levels of transmission lower and lower. But back then in 2007, 2008, 2009, with a group at UCSF, the division decided to take on the use of TDF as prevention in the IPREX trial. That trial was done and completed, and showed to be, just as a bulk aggregate, about 40% effective. But through very careful analysis of samples from study participants, it was demonstrated that if there was detectable blood levels of tenofovir in the blood, that the level of transmission was incredibly low. So that's one of the beauty, it's one of these interesting chemical anomalies, that TDF actually concentrates in red cells. So you can take a dried blood spot from somebody and tell approximately when they took their last dose of TDF and how frequently. And that discovery really was a game changer in demonstrating that if people took their pills, again, we're back to a question of adherence. If people take their pills and stay on with PrEP, it's quite effective. That was ultimately was one of the trials that was used by the US FDA to license the use of TDF for HIV prevention in men. So there's another example. We had treatment as prevention, we've talked about. What we've seen is TDF is not for everybody. We're really good at starting people on PrEP, not so good at maintaining people on PrEP. So we pursued a couple different angles. We tried a microbicide angle, and ultimately had a ring that made it with a Janssen drug called dapivirine. That ring has now received marketing approval in several countries in Africa. But the other angle we took was long acting. Okay. Could we make an injectable that was in many ways a little bit like the injectable birth control, that you would give it, you set it and forget it, come back in 2 to 3 months, get another shot, and see what you can do. So the trial that, again, the HPTN, the HIV Prevention Trials Network performed was a type of design called double-blind, double-dummy. Okay. So everybody took pills and got shots. Half the people got active pills, which was TDF. Half the people got injections of a drug called cabotegravir that is in a long-acting formulation. And the results of that study, which was completed during the pandemic. So in the midst of running these two large trials, one in men and one in women, the global pandemic for coronavirus hit. We were able to continue those studies and complete them during the pandemic, and it demonstrated that CAB-LA was superior to tenofovir in both men and women for HIV prevention. And that finding remains an incredible bellwether for next steps. So what we have seen, similar to with therapy, is that once you have an indication that looks like this is going to be a game changer, companies jump in. Right. And other companies are now working at other long-acting agents that are longer than 2 to 3 months. Some may go 6 months. There are other strategies being considered. So it's all about the innovation that we can bring, the proof of concept, even if it means getting a product all the way through to licensure. And then having the private sector come in and continue the innovation, because essentially the path is clear. It becomes an engineering question. It's no longer a matter of fundamental discovery. And so I think that's the role the division has truly played, as being, we have a concept, a scientific concept, reduce it to practice and test it through clinical trials. And then we can take that information. The company that worked with us goes out and makes their product. But the next round of companies come in and say, well, I've got a better idea. I can build a better mousetrap. And they go off and they do so. And pretty soon the injections are lasting once every 6 months. And somebody else will come and say, well, I can do better than that. I can make a monoclonal antibody or a set of monoclonal antibodies that are safer and easier to deliver. And they'll last 6 months too. And that's the kind of waves of innovation we continue to hope to spark. But in the meantime, each of these studies tells us something about what is needed to prevent HIV infection. And it all comes back to two things: either reduce the amount of virus that is coming into the exposed individual, or make it so the cells in that individual who's being exposed to virus, either the cells are resistant or cannot be infected by that virus. And that's where the CCR5, the Delta 32 CCR5 homozygotes came in, because they were completely resistant to R5 viruses. We know that monoclonal antibodies at high enough concentrations can block acquisition. We know that drugs can block acquisition. So we have a model that, if you can figure out how to prevent that first couple steps of HIV acquisition, which is probably the most vulnerable part of the HIV life cycle, is that from virus to where the first cell gets infected, and whether it's that first cell that makes a bloom of virus that then spreads, how many rounds of replication can you go before you have passed the point of no return? We don't have an answer for that, but we know if we can prevent very early steps, that we can prevent HIV infection. So now to segue to your next question, so how does this apply to vaccines? And the way this applies to vaccines is that is what a vaccine needs to do. It needs to prevent the establishment of infection. No vaccine ever licensed does that. So this is the challenge we face, is we would like to make a vaccine that does something no other vaccine has been able to do. And we have tried for decades and not succeeded. But there is still reason for optimism. And I think that is what I want to leave your audience with today. It's like, we have learned so much, and we've really narrowed down the target. We thought we had an approach with the Janssen vaccine, the Ad 26 approach, but the type of T cell response we were inducing didn't kill.
Okay.
So it's not enough just to have a good T cell response,
Right.
making gamma interferon and IL-2 and all these other things. You actually have to kill. And so this is as we go forward, what we need to do is treat this not so much like a war theme, but more like a symphony, where we have to have all the different parts of the human immune system come together in tune on the same sheet of music to prevent HIV from establishing a toehold. There are cofactors and other things which make it easier or harder for the virus to do so. But to me, so you mentioned earlier I was trained as a biophysicist. To me, it's about reduction to a principle that can be simply tested in a way that gives you clarity. Because you can do all the thought, you do great thought experiments, but then can you make that actionable in a clinical trial or experiments in non-human primates or in some systems that give you a sense that you are on the right path?
Yeah.
Because to me, that's the best part of the biological sciences, when you marry that kind of focused, razor-sharp question with the messiness of human biology to get such a clear answer. And that's what we did with 052. It wasn't about testing delivery of drugs. It was about maintaining people who were virally suppressed.
Yep.
And that was what was being tested. It wasn't drugs, it was viral suppression. It was the full act of the drugs. So you have to be crystal clear in your thought about what you're evaluating. So, and I think that, HIV is a particularly challenging virus to work on because, basically maybe there's now one person or two of the 80 million people who have ever acquired HIV that may have spontaneously cleared their infection since 1980. I mean, so that's not very good. That's not very good numbers for, like with coronavirus. Yes, we have a high death rate in the order of maybe as high as 2% in certain populations, but there's convalescence.
Right.
People do recover. We know we can make an immune response which mitigates pathology and disease. HIV, we don't have that blueprint. So we need a new blueprint. And my blueprint is preventing that initial first step. And we know from experiments that monoclonal antibodies at high concentration can do so.
Yeah.
So, but I personally think that it's going to need us to make a vaccine that not only triggers broad neutralizing monoclonal antibodies, but also a protective T cell response. And the two of them together can do the job. There are actually some animal model experiments from Rama Amara at Emory at the Yerkes Primate Center, where they have strong T cell response and add monoclonals in and show that there's an additive effect of the exogenously added monoclonals in terms of protection. So I think the pieces are there. It's about us now building the human vaccines that can do what we are. The engineering problem we have to solve is making the vaccines so they're deliverable without, starting somebody's 9 years old and give 6 years worth of shots. We need to reduce it to practice to a point where it's a testable hypothesis for both a T cell response and a monoclonal antibody response. And then we can have the potential of a vaccine that may really, really work. The work that was done on the monoclonals in identifying those epitopes, I think, is one of the other great stories in HIV.
So yeah, so we're now. I appreciate you taking us here because again, it segues perfectly. We in this symphony that you mentioned, this HIV vaccine symphony. So we require a strong public-private partnership component. We require this combination, this durable T cell response with the monoclonal response. We bring it all together. We are now in the, here in 2023 with the, well, you have a couple of things in the portfolio, but obviously the HVTN 142 trial was just everywhere in the news, which again incorporates this VIR 1388 vaccine. You guys, Gates Foundation, Vir Biotechnology. So obviously putting it all together. And you have other things in the portfolio. Obviously, this is and you can walk us through sort of CMV vector and how this particular structure is unique and interesting. But you have other things in the portfolio, the monoclonal antibody, VRC01, you have some mRNA vaccine stuff going on. Talk a little bit about the HIV vaccine symphony now, if you would, in that particular aspect.
So whenever a door closes, it is in some ways a gift, because that is a dead end and it's not worth going down. And so the door that remains open is this idea of exploring how we can teach the human body to reproducibly make the antibodies, which we refer to colloquially as the broad neutralizing monoclonals. And there are several categories of those in terms of binding sites on the HIV envelope. We have the CD4 binding site. We have a region called EMPER, which is part of the entry mechanisms. It's right at the interface between the virus and the lipid surface of the virus. We have the V2 loop, and we have the something called the glycan patch. Okay. With those, can we make immunogens that will reproducibly trigger responses that are broadly neutralizing with either starting by germline activation or some mechanism to get those antibodies started and through subsequent vaccinations matured to where they're being made at a rate and speed and number that they could be protective. We have several groups working on that. They're making really good progress. One of the breakthroughs has been animal models, particularly mice that have been genetically engineered. So they're almost like light switches. If the immunogen works, it's so clear that it's working in these mice. And if it's not working very well, it's very clear. So that's been a breakthrough because, how do you test a vaccine? But again, I think this is the brilliance of people like Fred Alt and Facundo de Batista, who've been able to build these mice that can really be used to streamline the evaluation.
Yep.
Monoclonal, so the way we make the vaccines and test them, messenger RNA is an incredibly powerful platform because essentially you can make any sequence, encapsulate it in the lipid nanoparticle and test it quickly. You don't have to purify it, the protein, you don't have to go through all these steps. And so in a non-clinical, and by that I mean not going into people, you can iterate with messenger RNA and test lots of constructs either in the mice or in non-human primates and really narrow down and select the best sets of immunogens. So that's the work that's going on right now, targeting those different spots on the envelope. Mm-hmm. CD4 binding site, EMPER, V2, V3, the glycan patch, those are all targets that are in play. There's another site that the NIH VRC is working on called the fusion peptide, which is a really interesting one. So I would say we've got several shots on goal. We need a couple of these to work in order for us to start thinking about testing in efficacy studies a vaccine that induces broad neutralizing monoclonal antibodies. Mm-hmm. That said, that's only half of the symphony. The other half is the T cell response.
Right.
And Louis Picker had a better idea years ago, and using the rhesus CMV, that's a type of monkey that has its own specific CMV, he demonstrated that he could make a vaccine that would induce not the usual suspect CD8 cells, but using a different something called HLA-E, which people hadn't really thought about in this field or in any field, frankly. And Lewis demonstrated that this vaccine could be incredibly effective to the level of about 55 to 60%. Interestingly, this also works really well as a TB vaccine. As a delivery. So as a platform, CMV could be a really important platform for not only the chronic disease of HIV, but also for human tuberculosis. So that is one of the other reasons we're really interested in this, because there's billions of people around, I'm not being too hyperbolic, people living with TB. And so to be able to really have an impact using a platform on HIV and TB is just such a wonderful vision of where this whole thing can go. So back to Vir, we're testing their version of their HIV vaccine in phase 1/2 clinical trial with HVTN, and we'll see where that goes. But again, I think this is an approach to T-cell vaccines. There are other approaches as well, using other viral vectors. And so I think that we're going to continue to see this growth of the T-cell response becoming more and more important, because I think everybody's going to realize we have to have the bass, we have to have the flutes, we have to have the violins, and we still need a conductor to direct all this. And the conductor is, I think, the most underappreciated part of this whole thing, and that is the CD4 positive T cell. That is the target of HIV. So what HIV does is it goes in and infects the activated CD4 cells and kills them. And so it's literally, it's like it cuts off the head of the conductor almost immediately by decapitating and eliminating probably the most important subset of CD4 cells. So we're dealing with a really nefarious pathogen here that in some ways, once those cells are gone, they may never be able to be reformed because those were generated after years of growth. I mean, T cells that were formed when you were a baby are part of your mature resting T cell response. If those get killed, they're not going to be replaced. So that's what the kind of thing we're dealing with. So moving forward, we need to make progress, whether it's the Vir CMV platform or other platforms, testing ways of inducing very potent CD8s and other modes of cell-mediated immunity. We have the monoclonal antibodies, but these are like two different machines moving down the road. We're going to have to come to a point where we start doing mix and match experiments. We're going to have to get company A to work with company B. There's going to be all this parochialism. We will get through that because it's in everybody's best interest to. We've been down this road before where we tested the CD8 concept and it didn't work. We've tested an antibody concept. It didn't work. We have to get to a point where the community, and by community, I mean not just academic researchers and the people at risk of acquiring HIV, but the industrial community, the pharmaceutical community also has to come together to realize that this is a bigger problem than any one company can settle on their own. Yeah. It has to come together as an integrated whole.
And Carl, as part of that integrated whole, one of the, there's another piece to all this, which has to do just with the way you structure these clinical studies ultimately, and you sometimes have to get creative. And I think I'm here in Philly, and I've had the local vaccine luminary Stan Plotkin on the show a couple of times. He's not related to HIV, but talking about things like challenge studies, preparing coronavirus vaccines, and things of that nature. What what should we know about innovation in clinical trial models per this part of your portfolio? Because I know that you published on some different things, maybe some of, in terms of adaptive trial design, maybe that hasn't really taken off, but other areas like control groups and the use of the control group. What should we know that's happening in clinical trial innovation?
Well, I think one of the areas that I think is underappreciated is if you think about the person who is already living with HIV and is virally suppressed.
Yeah.
In some ways, if you take these vaccines, particularly the ones that induce monoclonal antibodies, and immunize them, can you raise the level of antibodies in their blood to a detectable and maybe therapeutic level? And then using a playbook, part of the playbook of the cure research field, take them through what we call a structured treatment interruption and see what happens when you've raised the level of antibody in the blood of a person by vaccination. Are they protected from rebound?
Mm-hmm.
So again, if it's almost like you're asking, does the internal virus remain sensitive to those antibodies? And by all rights, that's a testable hypothesis there, is to isolate the virus from the individual and test that. And that, I think, is going to be a major way that we evaluate some of these immunogens to get a sense that these things truly can work. Because we already know from work by Michelle Nussensweig's group and others that if you put antibodies in people living with HIV and take them through a treatment interruption, many of these people are protected from rebound. And I think that is going to be a very if you think about what that means, is they are maintaining a low or undetectable level of HIV in their bloodstream, even though they are no longer taking antiretrovirals and all the protection is coming from the antibodies. And does that antibody response then promote almost a second wave of immune response against that virus that's emerging? The answer is, it looks like maybe that's true. The same thing can be done on the T cell side. Can we boost the T cell responses? So I think what we're going to see is the continued blurring of the line between treatment and prevention, as we've seen with the drugs. But now we're going to be blurring the line even more between vaccine, therapeutic vaccine, and cure and preventive vaccine. So bringing the field essentially, in physics, they talk about a unified field theory. Because there's one equation that can explain all states of matter for everything. It's been a question for forever and ever. In some ways, this is where we're headed in HIV, is this integration of it's all about the virus and how it's physiologically controlled within the person. And it doesn't matter if it's a new infection coming from the outside or if it's inside and emerging in a lymph node where it's essentially part of a rebound. That kind of an approach. Then we can talk about some interesting test of concept designs to test, because at the same time, we're going to have very potent antiretrovirals that'll be injectable and possibly delivered by a skin patch or other mechanism that will last 6 months. So we're going to have some sort of study design that incorporates a standard of prevention with an HIV vaccine. But if we have good evidence that the HIV vaccine has a high probability of working through one of these other systems, we can have a system similar to what we had with the HPTN 083 and 084, the cabotegravir studies. We have active control plus vaccine, and it becomes double-blind, double-dummy, or something along those lines. I think that this is where we are going to need extensive community consultation. We're going to need buy-in from host country governments.
Yep.
And at the same time, you could even think of a design where you took maybe we'll get to a place where we're doing designs where you take an entire village or population and provide the vaccine or provide other prevention methods, and then do some sort of additional measurements and switch. But I think we can get to a point where we can safely evaluate these vaccines. But I think the bar is going to be higher in the future for starting an efficacy trial. And I think it's going to require that kind of either test of concept in really true non-human primate models, or in the kind of studies I was discussing with you that are looking at vaccines to prevent rebound in people who are virally suppressed and are willing to stop their antiviral medication. Mm-hmm. So I think we rely on ingenuity. We rely on design and other strategies that are unique and thoughtful, and can really then change how we approach things. But again, it's going to be through conversation, through discussion with, Stan has been part of our fabric, helping us on the HIV front, Stan Plotkin, for as long as he's been in vaccines, which is longer than I've been in vaccines, which is saying a heck of a lot. But people like Stan, really still have a lot to contribute, and we're going to need people who've seen it all, done it all, and still have great ideas. We need new ideas. We need a community of next generation researchers. We need active, engaged community members to help us stay focused on the questions that are of most interest and importance to the community directly. At the same time, really remember that what we're about is coming up with the best tools to prevent HIV infection, as well as tools to help cure HIV infection. Mm-hmm. And the fact that these two can come together in a synergistic way, I think, is going to be incredibly powerful.
Carl, in December of last year, 2022, you, Anthony Fauci, and some other luminaries in space in the Journal of the International AIDS Society published the paper 40 Years of Investment in HIV Research, Progress Towards Ending the HIV Pandemic and Preparation for Future Pandemics. And in this paper, you talk about PEPFAR, which you mentioned at the beginning of the show. Obviously, a lot of what we're talking about right now could not have been achieved just sort of with the older model of short-term grants, and that this, as you mentioned, this collaborative science, cooperative research enterprise model, and big funding behind it is really what got us here. As we're on the 20th anniversary now of PEPFAR, we just had Ambassador Kengasong on. There's political stuff happening around PEPFAR as we speak. Take us, I mean, give us your perspective on where this needs to be going in terms of, obviously, this moonshot type funding for this next phase of what's happening in your lab and your organization, but also important learnings in terms of, if tomorrow you do solve HIV and we're not worried about this thing anymore, are there things we can learn from this model for Alzheimer's disease, for pancreatic cancer, whatever the case may be, and how to get some of this stuff done as we move forward?
Absolutely. And I think the example for other chronic diseases that I can give you is cardiovascular disease.
Okay.
So we have just completed a study called Reprieve that started over 8-10 years ago, where there's been this fact that people who are living with HIV as they get a little older, even though they're on medication and virally suppressed, have a 50 to 100% increased risk of major cardiac events. That's not a trivial thing. And it is in some ways more prevalent in women than in men. And it is also globally not evenly distributed. So working with, and this gets back to the whole notion of collaboration. Okay. We, in collaboration with the Heart, Lung, and Blood Institute here at NIH, we had the infrastructure for trials that could be done internationally. They had the cardiovascular expertise, and along with the Office of AIDS Research, we supported this REPRIEVE trial. And it had enrolled well over 7,000 people, men and women, well-balanced, ethnically well-balanced, racially well-balanced, and it was profound. We didn't expect it. The trial was stopped by the Data Safety Monitoring Board because we had hit a level of endpoint that was higher effect size than we ever thought we could ever see. And it's because we were using a risk calculator that would determine potential risk. So we used people that were not at their major risk factor was living with HIV. There were people who obviously needed to be on a statin because that was what we were testing. So the question was, can the addition of a statin, one pill once a day, to somebody who's already virally suppressed have an impact on their cardiovascular risk? We enrolled a population from the ages 40 and up, which is actually young for cardiovascular disease, but it was a profound effect. There was excess protection than we had ever forecast. But that's the kind of thing that we can do when you collaborate and you have infrastructure like this. You can ask big, important questions and answer them. So for coronavirus, we were there and we had a very good distribution of global research sites. So when there needed to be testing of the coronavirus vaccines, our sites were available. Mm-hmm. We were there for testing the antivirals, and there's still a study of a drug that is made by Shionogi Corporation, coronavirus protease inhibitor that is ongoing. So, the point of the article was we have infrastructure that we are already keeping warm because it's doing the business of figuring out how to better treat and prevent HIV infection. We've already demonstrated that when a global pandemic hits, we are in enough places that we can literally turn on a dime and be ready within a very short period of time to implement vaccine trials, therapeutic trials, prevention studies with monoclonal antibodies as needed. So we don't need to go out and make warm infrastructure. Yeah. We already have that within the Institute. And furthermore, it's incredibly well trained on how to perform randomized controlled clinical trials, because that is our business. So I think it was an important article that we were able to write with Dr. Fauci right at the end of his tenure as NIAID director, because it really does, there's a big debate right now. How are you going to maintain a warm base? And the point is, we already are maintaining a warm base because the warm base is fully employed, but the warm base can switch and talk and chew gum walk and chew gum at the same time, because we were able to complete the 083 and 084, the injectable cabotegravir trials at the same time we were also running these other studies. So right. It really stretched people and strained the capacity, but that's what it's about, is right. When human existence is under existential threat, you respond. And the networks and the people in the networks, we owe a huge debt of gratitude for the leadership and the quality of the work that was done to help us get the vaccines evaluated, to get the medicines evaluated, and to help us truly understand what COVID is about.
Well, while we have you, I noticed this year you've, in addition to everything that's going on at the organization, you've been at the conference on retroviruses and opportunistic infections, which was earlier in the year, the International AIDS Society conference just a couple of months ago. Anything else coming up in terms of public-facing stuff for the rest of 2023 as we get into 2024 that we should know about, places that we can run into you, meet you, listen to you, talk about these issues? Anything else I missed, please?
Well, I think there is movement. There's movement on TB that I think is really, really important, not just for this division, but I'm not the face of that work. But I think as a division, really being able to have profound impact on TB through treatment prevention and ultimately evaluating additional TB vaccines.
Mm-hmm.
I think it's absolutely critical. In terms of the work that I'm focused on now, I am still focused to a certain extent on better medicines for coronavirus disease as well.
Okay.
There's a program called APP that we're involved with, which is working with centers around the country to make additional medications for the purpose of preventing not just coronaviruses, but other potential pathogens of pandemic potential. So really get ready for the next pandemic.
Yeah.
Both at the level of what are the tools we're going to have? Can we have platforms like mRNA vaccines designed for a Bunia virus, for a hantavirus, for all these other viruses that have pandemic potential? Chances are there may be another coronavirus, but it may be different enough that the drugs and vaccines we have aren't exactly aligned, but because it's a coronavirus, we will know how to do that better in the case of the next pandemic. We also have to do something about making sure that people understand that this is a real pandemic and that coronavirus is a real disease. There's these very depressing statistics about the deaths that have occurred between January 1st and July 1st of this year. The vast majority of people who were hospitalized were over 65. The vast majority of people who died were over 65 and unvaccinated. And all of these the sad thing is all of these remain preventable deaths through figuring out how we're going to battle disinformation so that we can get to a place where we have diversity, equity, inclusion, and accessibility for all Americans. And that includes access to healthcare, access to housing, access to food, all of these essentials that should be the rights of being a citizen, but they're not. So that's about as political as I can get.
Yeah, I mean, it's an amazing journey, Carl. It's just to hear you talk about it and to see where we potentially can go with this. Obviously, the TB topic, is another one that we've touched on. And again, thinking back to the fact that the BCG vaccine was before penicillin, puts that all in some perspective. But if we can catch up on some of these simultaneously while you continue this amazing, movement.
Well, there's been some really good articles recently about a vaccine that the Gates Foundation is taking forward for TB that I think is worthy of some attention. So I think that it's taken a while for them to get it to a right. They're taking the appropriate steps to make sure that when that vaccine is tested, it's tested appropriately. And I fully applaud and support what they are doing in that space.
Absolutely. Needless to say, I'm a big fan, Carl. I'm rooting you and the whole organization on as you continue to move down this path. And again, for everybody that is going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel, you've been spending time with Dr. Carl Dieffenbach, Director of the Division of AIDS, National Institute of Allergy and Infectious Diseases at the U.S. National Institutes of Health. Carl, again, I want to thank you for taking the time out of your schedule to come talk to us for a little while about everything you've been doing. Thank you for continuing to do it. And as we like to say on our show here, thanks for helping to create a better tomorrow for millions of people out there via what you do. A really great story and wishing you all the best with it.
Thanks for having me on. It's been a lot of fun.